The lymphatic system has four functions. The first function is fluid recovery. Fluid from the capillaries can accumulate in the tissue spaces. This is prevented by the lymphatic system that picks up the excess fluid and returns it to the general bloodstream. The second function is absorption of fats. Lymphatic capillaries absorb fats at the small intestine. The third function is the production of lymphocytes. There are two types of lymphocytes, T lymphocytes and B lymphocytes. The organs of the lymphatic system produce these lymphocytes. The last function is the storage and mobilization of lymphocytes when there is an infection. Lymphocytes are stored in the lymph nodes, and when there is an infection, they must be mobilized and sent into the general circulation. Let's take a closer look at fluid recovery. Here's a blood capillary. The red side is the arterial end and the blue side is the venous end. Water comes out at the arterial end and water returns to the blood capillary at the venous end. Inside the blood capillary, the fluid is referred to as plasma. Even though there is movement of water in and out of the capillary, a certain amount of fluid ends up in the tissue space. This excess tissue fluid can then be picked up by the lymphatic system. Once it enters the lymphatic capillary, this fluid is now referred to as lymph. The lymph then travels into lymphatic vessels. There is a lymphatic vessel carrying the lymph into the lymph node and a lymphatic vessel carrying the lymph out of the lymph node. So the area that is expanded in the center is the lymph node. From the lymphatic vessels, the lymph then flows into the lymphatic trunks. From the lymphatic trunks, the lymph flows into a lymphatic duct. The lymphatic ducts then send the lymph into the subclavian vein. Here, the fluid is now referred to as plasma. And from the subclavian vein, the fluid returns to the heart. This is how the lymphatic system constantly picks up excess fluid and returns it to the bloodstream. There are four major lymphatic organs. We'll start with the lymph node. There are thousands of lymph nodes all across the lymphatic system. When there is an infection, lymph that contains the pathogen can enter the lymph node. And when this happens, the cells inside the lymph node sense that there is something wrong and they become activated. So the function of the lymph node is to store lymphocytes and other white blood cells and mobilize them in times of infection. The second major organ of the lymphatic system is the bone marrow. Bone marrow is found in the joints, rib cage, shoulder girdle, and hip girdle. The bone marrow is the place where all blood cells are made. This includes white blood cells, red blood cells, and platelets. The white blood cells that are important in the lymphatic system, such as macrophages, neutrophils, lymphocytes, T lymphocytes and B lymphocytes, all originate in the bone marrow. The B lymphocytes also mature in the bone marrow. The T lymphocytes, on the other hand, need the thymus gland, which is the third organ of the lymphatic system, for maturation. So the T lymphocytes mature in the thymus gland because the thymus gland releases a hormone called thymosin. The last organ is the spleen. The spleen stores all the formed elements, white blood cells, red blood cells, and platelets. Let's look at how the body defends itself against pathogens. We have three lines of defense: the first line of defense, the second line of defense, and the third line of defense. The first line of defense is what the pathogen encounters first, like your skin, hair, mucous membranes. Those would be like the first line of defense. You want to imagine the first line of defense as your basic security that you see in any organization, like campus security, for example, would be your first line of defense. The second line of defense is what the pathogen encounters after it's gone past the first line of defense. So this would be a little bit more sophisticated. So that might be your police force, could be your second line of defense. The third line of defense is far more specific and it's the most sophisticated kind of defense, and this is like the special forces in the army. So the first line of defense is non-specific. This means that it protects the body against all pathogens. The same goes for the second line of defense, whereas the third line of defense is highly specific. For example, antibodies produced against the COVID-19 virus would be an example of the third line of defense. Let's take a closer look at the first line of defense. The first line of defense are barriers that are already there in the body and is the first thing that the pathogen encounters. So it is difficult for the pathogen to penetrate these barriers and enter the body. So we have three types of barriers. We have physical barriers such as skin, hair, and mucous membranes. We have chemical barriers such as hydrochloric acid, lysozyme, and vaginal secretions that are acidic. And we have physiological barriers such as coughing, sneezing, vomiting, and diarrhea. So let's take a look at each one of these. The physical barriers, we start with the skin. The skin is what the pathogen first encounters, so our skin is our first barrier. The second barrier is hair. The third barrier are mucous membranes. We have 4 tracts in the body that point to the outside, that are open to the outside world. Those are the digestive tract, the respiratory tract, the urinary tract, and the reproductive tract. All these tracts open to the outside world, which means we are more susceptible to pathogens entering. So the mucous membranes release mucus and keep these tracts very slippery, not allowing the pathogen to penetrate the deeper tissues. The physiological barriers are obvious. Coughing, sneezing remove pathogens from our respiratory tract. Vomiting and diarrhea remove pathogens from our digestive tract. They might be unpleasant, but they are there for a reason. They benefit us by protecting us against the pathogen. Chemical barriers, we have many chemicals. HCl in the stomach, lysozyme, which is an enzyme that is found in saliva and tears and nasal secretions. They deter bacterial growth. They basically kill bacteria. Sebum, which is oil that comes along with your hair follicle, also deters bacterial growth. And vaginal secretions, which are acidic, can kill bacteria. The second line of defense is more sophisticated than the first line of defense because the immune system should actually do something to deal with the pathogens that have penetrated the first line of defense. You want to imagine the second line of defense as being a combination of the FBI and the armed forces involved in combat or rescue. We're going to classify the second line of defense into 3 types: protective processes, protective proteins, and protective cells. So we'll start with the protective processes. There are 2 types of protective processes that we will look at. One of them is inflammation, and the second is fever. Both inflammation and fever are beneficial to the body unless they go out of control. The second is protective proteins. Let's look at 2 types of protective proteins. One of them is the interferon, which is an antiviral protein, which we will look at closely in a bit. The second is a complement protein. Complement proteins are a set of proteins floating around in the blood waiting to be activated by the presence of a pathogen. The third group is the protective cells. We will take a look at two major protective cells at the second line of defense, and they are the natural killer cells and the phagocytes. The natural killer cell is a kind of lymphocyte, and the phagocyte is the macrophage or monocyte. Let's imagine that the skin has been penetrated by a sharp object, bringing with it some bacteria that we see in blue. The bacteria has now penetrated the epidermis of the skin and it is in the dermis. The injured tissue produces alarm chemicals shown in yellow, and these alarm chemicals stimulate a cell called a mast cell that is shown in blue. The mast cell is a cousin of the basophil, and we know that basophil releases histamine, and histamines make capillaries leaky. So that's what we see here. The histamine from the mast cell makes the capillaries leaky so that neutrophils and macrophages can squeeze through the capillary and arrive at the site of injury. So that's what we see here, the neutrophils and macrophages arriving at the sites of injury. So let's go over the steps one more time. The first step is that the tissue is injured by a sharp object and bacteria enters the skin and reaches the dermis. The injured tissue now produces a vast variety of chemicals, one of which stimulates the mast cell. So alarm chemicals are released by the injured tissue. The third step is that mast cells are stimulated. Mast cells are cousins of basophil. They are like the basophil and they release histamine. And histamine is known to make capillaries leaky so that more things can come out of the capillary. Once the capillaries are made leaky, the white blood cells can now squeeze out of them. That's step 4. The neutrophils and the macrophages are attracted to the site of injury, and so they move to that area and then they squeeze out of the capillaries and arrive at the site of injury. Step 5 is that the neutrophils and macrophages kill the bacteria via phagocytosis. Fever is an indicator that there is an infection. However, fever is actually beneficial because it is a process that allows us to fight off infection. Our body temperature is set around 37 degrees Celsius. We call this the set point, and it is registered in the hypothalamus. Even though we say that 37 degrees is the set point, our body temperature tends to go a little bit higher and a little bit lower than 37 degrees. We call this an oscillation. We say that it oscillates around the set point. When there is an infection, however, the set point goes way up to 39 degrees Celsius. So now the hypothalamus registers the set point, new set point, at 39 degrees Celsius, and it stays up there until the infection is over. Once the pathogen numbers go down, the set point goes back to 37 degrees Celsius and we return to the oscillating 37 degrees Celsius. Now let's look at these stages one by one. At the onset of an infection, certain lymphocytes release a chemical known as a pyrogen. Pyro means fire. So this pyrogen helps to raise the set point from 37 to 39 degrees Celsius. So the new set point is now 39 degrees Celsius. The body needs to now achieve that new set point, and while it does that, you're going to feel cold, shivery, and a little uncomfortable. Once we reach the set point, the new set point, which is 39 degrees Celsius, the body temperature will oscillate right around the 39 degrees Celsius. This continues until the pathogens have been destroyed. Number 4 represents the end of the infection. Once there is no more pathogen, there is no more pyrogen release from the lymphocytes, and so the set point goes back to normal. Let's recap these steps one by one. Number 1, there is an infection and there is a pyrogen secretion. 2 is the temperature set point going up. So it goes to a new set point. Stage 3 is the fever set point. We have arrived at the fever set point and you're maintaining this set point. 4, the infection has ended and the set point goes back down. Step 5 is the body temperature going back to normal, returning to the original set point. Now for the benefits of fever. Pathogens in general do not like high temperatures. They don't do very well at high temperatures. So fever helps to retard bacterial growth. It also slows down viral replication. The second benefit is that high temperatures raise our metabolic rate, and in doing so, they also intensify our immune reactions, so we are better able to fight off the pathogen. So fever can actually be a good thing. A natural killer cell is a special type of lymphocyte that has the ability to carry out immunological surveillance. These cells travel through the bloodstream looking for cancer cells and virus-infected cells. Unhealthy cells such as these look quite different from the normal cells. These enemy cells carry special receptors on their cell surface that can be recognized by the natural killer cell. So let's take a look at how a natural killer cell destroys the enemy cell. The natural killer cell is shown here in pink and the enemy cell is shown in blue. The enemy cell could either be a cancer cell or a virus-infected cell. So when the natural killer cell comes in contact with the enemy cell, it releases proteins known as perforins. Perforate means to make a hole, and perforin is a protein that makes a hole. So that's exactly what it does. These proteins reach the enemy cell and make a hole in them. The hole is then used by the NK cell to introduce enzymes. These enzymes are small and granular, and so they are called granzymes. These granzymes make the enemy cell undergo apoptosis, meaning it causes the enemy cell to commit suicide. Once the enemy cell is destroyed, it is then cleaned up by a macrophage which carries out phagocytosis and clears the debris or remains of the destroyed enemy cell. Now let's see how a phagocyte destroys a pathogen and presents its antigens to more sophisticated cells of the immune system. So here is the pathogen in yellow and its associated antigens. You want to imagine antigens as if it were buttons on the coat of the pathogen. Here is the macrophage attempting to engulf the pathogen. We call this process endocytosis, or more specifically phagocytosis, because the cell is actually eating the pathogen. Once the pathogen is inside the cell, it is put into the lysosome. The lysosome then destroys the pathogen while keeping its antigens intact. These antigens, imagine they are buttons, they are worn by the phagocyte to say, I've just killed something and here are the buttons to show for it. These buttons are the antigens, and these antigens are presented to T lymphocytes, which are the most sophisticated type of white blood cell in the third line of defense. So the antigen presenting cell, or APC, is nothing but a phagocyte. Let's take a look at the protective protein called interferon. Interferon interferes with viral replication. In order to understand how interferon functions, we need to understand how viruses operate. A virus cannot multiply by itself. It must enter your cell and then use the resources in your cell in order to multiply. Once the virus multiplies, it then lyses the cell, the host cell, basically destroys it, and all the viruses infect the neighboring cells. This is how they operate. The interferon stops this from happening. So let's see how. We have two cells here. The cell on the left is being infected by a virus. The virus is shown in yellow. The virus does not enter the cell, but it introduces its genetic material. The genetic material could be either DNA or RNA. Once the genetic material enters, the cell responds by becoming activated and making a protein called interferon. Interferon is shown in pink. Once the interferon is made, it can now leave the cell via exocytosis and enter the neighboring cells. So interferon leaves the cell, reaches neighboring cells, and influences the neighboring cells and asks them to prepare for viral infection. So here's your uninfected cell. This cell does not know that there is a virus in the vicinity. Once the interferon signals this cell, the uninfected cell makes antiviral proteins called AVP. These antiviral proteins are like little weapons that are ready for a viral invasion. When the virus actually arrives and introduces its DNA or RNA, AVP blocks viral replication. If viruses cannot multiply, they cannot survive. So the AVP blocks the viral replication and basically stops viral infection. So let's recap. Interferon is a protein that is produced by an infected cell, and its job is to signal healthy cells in the neighborhood to prepare for viral invasion. The last component of the second line of defense are protective proteins called complement proteins. As their name indicates, complement proteins enhance or complete the action of other proteins and cells of the immune system. There are several kinds of complement proteins classified into many groups and subgroups. But what we're going to look at is how complement proteins in general influence or enhance immune functions. So let's take a look at the first function. Complement proteins help to enhance inflammation. As we know, inflammation is made possible by mast cells that release histamine and make capillaries leaky. And they also depend on neutrophils and macrophages that kill the bacteria. So the complement proteins stimulate the mast cells and intensify the action of neutrophils and macrophages. Together, they enhance the process of inflammation. The next function of complement proteins is to make pathogens more appetizing. Yes, you heard that right. The complement proteins act like curry powder on your food. Here's the pathogen shown in blue. The complement proteins shown in pink, they coat the pathogen as if you were to season your food, so that they would be more appetizing to the phagocytes. This actually has a name. We call this process opsonization, which means make the pathogen into food. And this really helps because the phagocytosis is intensified. The next function of the complement proteins is to make holes in the pathogen or any enemy cell. It is not unlike what happens with the perforins. Here, the complement proteins make multiple holes on the enemy cell, causing the enemy cells to be destroyed. We call this cytolysis. Cytos means cell, lysis means breakdown.